the hall. There's a very important day, I think, a landmark day in my opinion, where the current IIIM director and the two past directors are attending this meeting. And each of them are very, very different in their scientific accomplishments and have brought in very important, where came to the institute at very important juncture and were responsible for its transformation in very significant way. Dr. Handa came from the stream of pharmacy with a very strong knowledge of the Ayurvedic properties of plants and created a new walkway or a highway towards development of this area. Dr. Kazi, a biotechnologist, enzymologist and fermentation specialist, again brought in an absolute modernization and a new dimension into the whole thing. And Dr. Vishwakarma, who was a die-hard chemist and also a medicinal chemist, brought in the chemistry into the whole system, and thereby this transformation has been very healthy. And I don't know whether it was designed or ordained, but it just happened. And this really strengthened the institute to take a big walk towards the development of plant-based medicine at our country and in the global setting. India had taken some landmark decisions on some very good research which has moved the whole thing into entirely new orbit. Like one of the things which we started doing is cataloging our plants which were used in Ayurveda or which were somewhere documented, and the entire documentation occurred to protect the properties of this plant and also create a new dimension on intellectual property. This was a very important step, and thereby the knowledge which was very, very necessary, which was catalogued. The second major part which India took was the Indian biodiversity law. And Indian biodiversity law again protected the biodiversity of our major resources. And third one was a germplasm bank, and also creating proper tissue culture facilities around our country, so that some of the things which were in the verge of extinction or were exploited in a bad fashion were protected for the future. One more major thing came up, our environmental consciousness, so that we became friendly as far as various toxic heavy metals in the environment which used to ultimately seep into the plant system and create problems. They range from lead to mercury to cadmium to arsenic, you just name it and it were there. And we had laboratories which could assay it, and we laid down laws which protected it. And the same were for the microbial overload, microbial contaminants, and all other issues which made Indian products little disadvantaged in the global market. So this was again something which was very important and good. Then came another major effort of creating these plants into a format in which it became, growing these became profitable to farmers. So the agro-technologies were developed, genetic selection was developed, and many times these were very nicely created by putting in the modern science, because we had large number of plant molecular biology units which were coming up, and they contributed their mind. and the entire transcriptomics sometimes will work out. Like in the Ashwagandha network project of CSR, entire transcriptomic and genomic map has been done for this plant, and ultimately the 3 or 4 major projects were identified, and one of the products is now being taken up for development of a drug for the stroke. And here the whole standardization path was taken up, like the purification. None of the products were entirely pure, but the signatures, which will both chemical signatures and genetic signatures, as well as the active principle and product, was identified and is being now taken for development of the appropriate technologies, agro-technologies, and farmers are making a lot of money out of it, and it is seeping around the spectrum, which will be very beneficial for the both for the drug industry as well as the every stakeholder into the system. Because when we look at development of plant-based medicines, you need every stakeholder to be taken care of, otherwise it will not move and it will not develop. Then and another very major thinking came up, and here those who were practitioners of the Ayurvedic medicine, they became very much involved in the modern science and modern biology. In one of the CSR institute, that Institute of Integrated Biology, they worked out in the Ayurveda, people are divided into Kapha, Vata, Pitta, 3 categories. They found out that there is a distinctive genomic signature, genetic signature in these 3 categories. They have different kind of ability to respond to drugs as well as metabolization of the drugs. They have different SIP systems. And looking at that, it became very important that now you develop drugs and test drugs in these matrices. So testing methods came up. This was another major problem here. The testing laboratories for the plant-based medicines and plant-based products were not existing in India. There were very few testing laboratories which were there, and the high throughput screening of those testings were not available. Now they are also emerging in India, and this will be really very useful, because if we have a world standard testing laboratories, this cannot be done in the research laboratories, because research laboratories can't all the time work on the GLP principle, although they are GLP compliant, but creating a total GLP testing laboratory is entirely different ball game. And there, because in research the SOPs almost go on transformation and changing. So this particular lacunae is also getting filled up, and this is coming up at the moment in the private sector, but I wish that government sector also testing laboratories come up which help in this direction. Then I will finish by saying a few things, that some of the other concepts which are coming up with the plant-based drug development is the new technologies which have come up, and in these technologies now it is possible to purify majority of the components of the plant product in a very quick fashion, and then analyze their chemical nature, work out their structures, and sometimes you can take them into natural product development of the pure drugs by creating appropriate crystals and instrumentation, which ultimately helps. And libraries are emerging. I have seen 2 libraries emerging, one in the IICT, and maybe there is one library in IIM, that they became very useful resource standards for the future. Let me give you a few major developments which have occurred in this particular direction in India. Some of the very new and novel products which have come up, and some of them have moved towards clinical development, and few of them may have seen the market also. This one of the product which came up from the triple IIM was the piperine and the different analogues of piperine. This particular product, when added to a known molecule, it enhances its bioavailability. It also manages the SIPs so that the metabolism of the drug is altered. And thereby, some of the drugs which had a problem in getting appropriately absorbed and were interacting with the proteins and nucleic acids were ultimately made to work better. And rifampicin is one of the things which have now in the market with piperine, and piperine is being now tried for other products. So this was one major development which came up. In the triple IMM, another product which came up was in India was anti-diabetic drug through a plant which is known as Vijay. And ultimately, an attempt was made to synthesize the chemical analog of the Vijay, because in the Vijay you had to sacrifice tons of the plants, and it became impractical. And in this way, right from the Jambolina to many other substances, there were efforts to create anti-diabetic products, and some of those anti-diabetic products, a chemical analog has been synthesized, and some of them are present in a natural format. And this again went into the market. In the same way, in the Bacopa Munera, that is the Bacoside, there was a product which was developed and marketed by CDRI, and here again, it was the first time a proper standardization happened when there were multiple components which when you separated, the plant medicine didn't work in that particular fashion. This was difficult to standardize, but whole pharmacology, its pharmacy, its formulations were worked out so that these become standard product. Currently there is a product again which works on osteoporosis, and another product which is known as at the moment numbered as K38. After the Eli Lilly's product, it is one of the very strong bone formation and bone healing product. This is again come up in the same way. There is a product now which works on which works as an immunosuppressant, a very strong immunosuppressant, has been developed by another CSR scientist, because mostly in this area the CSR scientists have made a tremendous stride in this area, because CSIR took a very strong interest in these. And if I go on listing several anti-tubercular drugs, several drugs which act on the immunocompromised individuals, a large number of strong antioxidants, and have developed with creating a series of products which have ultimately made their mark. But I will finish with one major development. The CSR labs, few CSR labs combined to create an anti-malarial drug and its analog, that is artemisinin. It was very difficult in India because the artemisinin content in the artemisinin annua was poor in Indian culture systems. You had to get them from Vietnam, or you have to get them from Ural, or you have to get them from China. The same thing happened, those who were working with the glycyrrhiza glabra, the one in Iran was not that good, and one in Afghanistan was also not that good. So it was being imported from far off. The Indian glycyrrhiza glabra was also not good. So here, again the laboratories ultimately were able to synthesize the core of the artemisinin molecule, which were known as endoperoxide. So the India marketed the first endoperoxide in the world, and the Ranbaxy laboratories marketed that. And then in the Berkeley, Dr. Kassling's lab created the first synthetic molecules of artemisinic acid through synthetic biology. in an yeast system and later on in a bacterial system. The trick was to ultimately move the ring and close the ring so that you have, although here at the moment the major problem is creating a commercial grade system. So what I want to close with a comment that a tremendous developments are occurring in the area of the development of plant-based medicine. Completely new matrices, completely new technologies. We understand now much better how the protein systems in plant work. We now know better that a plant which cannot be grown in one environment can be transplanted and grown in an entirely different environment by creating systems which will make it grow, and creating technologies which will ultimately are expandable to commercial structures. And making and protecting IP in the plant-based medicine has also been a challenge, and that has also sorted out. And the technology has made it possible to identify the active principle as well as the other components which help in these areas. And the human system, interaction of these drugs with the human system is also now much well understood. So I think we are entering into an era where we can maximize all these efforts to create molecules which will be very novel and will move in three independent streams in their natural format through the Ayurveda principle. The new drugs which are made in Ayurveda principles were not in Ayurveda, and also moving them through the natural synthesis and creating synthetic partners which could be formulated appropriately to be used as drugs. And the regulation, as Dr. Vishwakarma said, is very, very important. It has to walk with you, with the scientist. If the regulation doesn't walk with the scientist, everything will be struck. And I must confess the regulation is very weak. We don't understand half of the regulation, and the regulators don't have the science base to ultimately work out the quick toxicological evaluation and the druggabilities and other systems which will take Ayurvedic medicine or plant-based medicine, even a natural product medicine, into a fast track. So here it is important that the scientists and the regulators work together to move this agenda forward. And I hope this sometimes happens in India. It is happening in China. It is happening in the, as Dr. Vishwakarma said, in the US FDA and other regulators. It's unfortunately not happening in India. And again in India, the regulation is far more fragmented. There is Ayush, there is a Central Research Institute, and I chair the scientific committee of the Food Safety Authority of India. They have also a nutraceutical stream where things are there, like there is a product known as pre-lipid. One doesn't know what it is, a drug or it is a chemical or what it is and where it should go. So there is we need to prevent fragmentation. If there is a fragmentation, we will be really stuck. So it becomes somebody's fancy where he puts his product for approvals. But the major approval is from the global community. I think that is the approval. It is not enough that we create a product and have approval in India and have approval nowhere else. It really doesn't make a sense. So with these points, thank you for inviting me. I will close my remarks.